All right, so I'm just going to repeat that what I said without recording is that I just don't want to rush through the process. So what I'm going to do is I'm going to present the part of the honors that they have done. Some of you guys are going to present that. And then what I'm going to do as a review, I'm going to just record after the class sitting in my office with a Camtasia where I can see the questions and the slides as well. And it would be much better experience. I know that you have to watch a video recording of 1.5 hour additional. So but you will be rewarded for that for sure. So the second announcement what I have, and I will just put it in an announcement as well if you will not see this video, that instead of doing the exam 6 o'clock to 11 o'clock on Thursday, you're coming back from the spring break. I'm just going to extend that. So we'll start from 6 o'clock, 6 a.m. on Thursday. It will be open all the way up to the weekend, which is the Sunday. Sunday, 11:59 p.m. So you have much more time to review the material. And if you need to ask questions, ask those. But just one thing, please remember that. I will answer your questions until 5:59 a.m. Thursday morning. But once the exams start, I have to have the ethical reason. I cannot discuss anything in there. So just make sure that you understand that it is not being mean, but the thing is that as the exam starts, I cannot do an office hours after that. So if you have any questions, even on Sundays, like in the week, I know it's in the weekend, but if you just want to do a one-to-one Zoom meeting or office hours, Sunday, Monday, Tuesday, anytime you can just ask. So it will be not in person, but that's it. We are going to talk about so everybody hear me good? Good. The coronavirus RNA synthesis. And let's start with some of the basic ideas. So this is overall structural overview. You have seen this picture many, many times. The main proteins are in there. One thing, just remember that here there is a protein written as HE or hemagglutinin. Hemagglutinin is actually HE. It's actually hemagglutinin esterase. Some of the coronaviruses have this protein on their membrane, on their envelope. But none of the SARS, like the original SARS, SARS-CoV-2, or MERS, feline coronaviruses, there are some infectious bronchitis viruses. None of them has a hemagglutinin in there. So that is one which is present in some of the viruses, in some of them they are not. But all of the viruses have the spike protein. That is the famous spike protein. It's a trimer. Then you have the membrane protein. You have the nucleoprotein N along with the approximately 30 kilobase of the plus-stranded RNA encapsulated with a nucleoprotein. Many of the plus-stranded RNA viruses have a naked RNA. This is an exception. Remember that. A retrovirus HIV is also an exception, a plus-stranded RNA encapsulated with that. So this is the overview of that, and you can see that how basically the structure is organized, and the membrane proteins and the nucleoproteins do interact, and you can see that there is an envelope glycoprotein E as well. These are present in everywhere. So again, just repeating: spike protein, membrane protein E, envelope glycoprotein, nucleoprotein RNA. These are all essential except the hemagglutinin. Overall structure. So this is just a recap of the single cell reproductive cycle. So after the virus enters, we have already covered that. Immediately, its plus RNA will be delivered into the cytoplasm, and it's going to synthesize these proteins, 1A, 1AB. These are the polyproteins. We're going to talk about this first. Then we are going to see that after proteolytic cleavage of the individual proteins, they will set up some factories to manufacture the components, the genome as well as the mRNAs, in the double membrane vesicle. They're going to make some subgenomic mRNAs, which are smaller than the genome, and also they're going to do the genome replication that all happens in these double membrane vesicles, which is derived from the Golgi, but made up by the scaffolding proteins of the virus. And then some of these proteins are going to go into RNA, and the proteins are going to be in the endoplasmic reticulum. And this is the end protein, which will encapsulate the produced RNA. And the ARGIC is the endoplasmic reticulum Golgi intermediate complex, where the virus is basically all together. But this last part is we are going to do it in module 3, so which is the next part. So we will just go up all the way up to this RNA synthesis. So very busy slide. So this is Jennifer.
Okay, so I talked about the proteolytic cleavage of 1A and 1AB into different non...
Hold the microphone.
Oh, sorry.
Because it's recording.
Okay. Is this okay? Oh. Okay. For my topic, I talked about the proteolytic cleavage of 1A and 1AB into non-structural proteins that are not RP. Maggie's going to talk about those in her next slide. So a general overview of the cleavage. I think Maggie also goes more into this in her slide. But the polypro cleaves at the 5 site of the ORF, and then it produces non-RDP, non-structural proteins that include NSP1 and NSP2, and then a bunch of different other ones as well. And I have all the structures and all their functions here that I'll talk about in a second. So proteolytic cleavage is just the cleavage of amino acid groups, if we're just to like define that. And it produces 17 non-structural proteins. It was like 12 to 16, whatever source I was looking at. And this is just a list of all of the different functions that they have. I'm not going to read them all because I think you guys can read them all, but we can go over the highlighted ones. So NSP3 is a large multidome protein, which includes the polypro protease and ubiquitin-like domains, and the poly-ADP ribosome binding in transmembrane domains. And then NSP4 and NSP6 have transmembrane scaffolding, and NSP5 codes for 3-CL-Pro, and which is the MPro, which is the main protease that does this whole cleavage. And the overall function of all of these is to work with RNA transcription. I think you can see that just from you've got like a helicase function, an exonuclease function, an endonuclease function, which are all things that we've seen before and know, but this is just one of the functions that's very necessary for correct replication of the coronavirus. It has been seen that you actually can go through replication without like cleaving at the 10-11 non-structural proteins, but it causes a bunch of different growth defects. So in order for your replication to actually be successful and for you to create a useful virus, you need to be able to cleave at this site and create all of these non-structural proteins that have very specific and necessary functions for RNA transcription.
Awesome. I mean, like that's great. That's what it is. And the 12 and the 16, that confusion is that what you see that in some of the cases, some of these proteins do not get cleaved, and the two parts remain attached to each other, and that's why it's a controversy that whether they are separate or not. So sometimes NSP7, 8 remains attached together. So that's why the number decreases.
Okay, so I did 1A and 1AB polyprotein proteolytic cleavage and generation of the RNA-dependent RNA polymerase. So the open reading frame encodes for MPro and 3-CL-Pro protease, which cut the open reading frame, and it also codes for the papin-like protease. And so the papin-like protease cleaves 5 sites on the open reading frame 1A to produce the non-structural proteins that are not included for RNA-dependent RNA polymerase. And then this 3-CL-Pro cleaves ORF1A at 17 different sites, and it produces the RNA-dependent RNA polymerase subtypes and other non-structural proteins. So the core of RNA-dependent RNA polymerase consists of 3 non-structural proteins, which include non-structural proteins 7, 8, and 12. And 12 is the POM region of the right-handed DRP, and it's the catalytic portion. And then NSP8 and NSP7 form a kind of like a compound, and they form the clamp of the complex, and they're the processivity factor, which allow like the RNA-dependent RNA polymerase to continue along the RNA. And the NSP8 is a primase as well, and there's 2 NSP8s and only 1 NSP7. And there's other non-structural proteins as well that associate with the RNA-dependent RNA polymerase for RNA synthesis, such as proofreading abilities and exonuclease abilities.
Awesome. Perfect. Any question, anybody?
Okay, so I had how the ribosome frame shifts to get to the second open reading frame. So this is with the genomic RNA. So when it first comes in and translates the non-structural proteins. So once it gets to the end of the first open reading frame, there's a slippery sequence followed by a pseudoknot structure. It's just like 3 stems, and it's pictured up there. So sometimes the ribosome can unwind it, like as it translates, and then it reaches the stop codon, but sometimes the pseudoknot like doesn't immediately unwind, which causes the ribosome to stall and slow down while it's on the slippery sequence, which then, because it's UA base pairing, it slips backwards and frame shifts in like the -1. So now like when it reads, it doesn't read the stop codon anymore, and it continues to translate farther into the second stop codon of the open reading frame, 1AB. And this is really important because the RNA-dependent RNA polymerase is in the second open reading frame. So if it didn't slip backwards, it wouldn't translate the RNA-dependent RNA polymerase.
Yeah, good job. Thank you very much. I'm like, very good. I mean, like, we talked about the pseudoknot structure in detail, and you can now really see that how these pseudoknot structures, I mean, like sometimes, so basically think about this way. So ribosome does have a ratcheting mechanism. So it just constantly feeding the mRNA. And now you have, like, suppose you're just pulling a rope through the hole, and then you have a bunch of like tangled up knot in there, and that's basically the pseudoknot. So it's trying to pull, and it cannot pull, sometimes breaking those hydrogen bonds. That you can see that there's some hydrogen bond in the stem loop 1 and 2, and then there's a stem loop 2 and 3, and then there's some between the top portion and the bottom portion. So you can see those lines, those are the hydrogen bonds. And then it just slips out the frame. Now, one of the questions sometimes somebody asked is that suppose instead of slipping 1 frame, if it slips 2 frames or 3 frames, what happens? So usually at that time, that may happen very well. It will probably encounter an abnormal stop codon, and it will just abort the synthesis in there, because whatever is going to synthesize, those will be junk proteins. It will not be, so this will be basically a nonsense protein synthesis. That's going to actually happen. And what we are going to do, that this process also precisely control how much of these protein can be produced for these purposes. So the last line, the frame-shifted translation attenuates or produce less protein production from the ARF1B, as they are not required in abundance. But we will go into more precise functioning of that as well. So here we are looking at, this is an extension of that I just added these slides. So here at the top you are seeing the slippery sequence, and it's a busy slide, but just try to just take home message is that, so ribosome is started to synthesize from the 1A. It goes all the way up to this, and then either it will stop, it can read that sequence, the stop codon right here, which is the UAA stop codon, or it may slip, and if it only slips, at that time it will make these polyproteins. So if it slips, then it will not read that stop codon. It will not be a stop codon anymore. So then it can synthesize that. So if you carefully look at that, what is happening here, so from 1 through 10, NSP 1 through 10, these are produced from 1A as well as 1B. So think about that, NSP 1 through 10 is produced in a larger amount, almost double the amount. And approximately, depending upon the viruses, the frame-shifted ribosome moves through the 1AB in between 25% to the 75% of the time. So it just depends how much it is. But what the take-home message is, your NSP 12, 13, 14, 15, and 16, these are produced in much, much less quantity compared to all other proteins. So you can easily see what happens here. You can see that right here is a core RDRP, which is a hollow enzyme. You have NSP 8 and 7, and this is NSP 12. And if you carefully look at that, NSP 12, you just need one molecule of this catalytic domain, and this NSP 7 and 8 together makes this hexadecameric complex, which is altogether 16 subunits. So it's a larger structure. It needs multiple subunits of the same protein. Obviously, it needs more. So this is basically the precise control, and you can easily see the accessory RNA synthesis like here. These are the accessory proteins which are also needed, but this is the precise control by the frame shifting by this pseudoknot. So that is the implication. And this is the detail of the pseudoknot, that's what you are seeing. And right here is the NSP3 that we're talking about. It has lots of different multi-domain structures. So this is your NSP3 that, who talked about that? Jennifer talked about that, yeah. All right, so after that, Jerry probably sent me an email. She is not here. Okay, all right. So after that, what we are going to talk about, let's go to the first one. See that this is the double membrane vesicle. Now you have all the proteins, non-structural proteins are made up. Some of these proteins already we know as noted as the membrane scaffolding protein. They actually modify the ER to make these double membrane vesicles, and they are going to set up the RNA synthesis, the transcription and the replication, mRNA synthesis and the replication inside these vesicles. So right here, you can see we are looking at this. So you synthesize the genomic RNA, making 1A, 1AB, and this is your replicase transcriptase complex. These are your RDRP and the associated proteins. And they set up the double membrane vesicles, which will synthesize the genomic RNA and the other more mRNAs as well. We are going to see that. So, and then you are going to do the assembly. Now, remember that as it is a plus strand RNA, your intermediate has to be a minus RNA for your mRNAs as well as your genome RNA. You have to first synthesize the minus RNA, which will act as a template. So what happens here, so the function of the double membrane vesicle is basically as soon as the mRNA comes into the cell, the genome RNA, it is considered as an mRNA. So there is no cellular signal that can recognize that this is a viral infection. But when the virus needs to synthesize that minus strand intermediate, that can be easily detected by the cell that this is of a viral origin or some other origin, and it will trigger the immune response. It will trigger interferons signaling. So when you do this inside the double membrane vesicle, the minus strands are inside those double membrane vesicles, and that is basically sequestered out or kept apart from the cellular detection. So that is the function that why they are inside these double membrane vesicles. So here we use two words. This is RTC, replicase transcriptase complex, which basically set up, do the mRNA synthesis and the genome replication inside those double membrane vesicles. All right, and these are some of the proteins. NSP346, we already talked about that these are the membrane scaffolding protein. They all have transmembrane domains. And if we just go to any of those structures, if you look at that, these are red lines, that what you see right here, these red lines in 3, 4, and 6 right here, these red lines are the hydrophobic sequences which inserts them in the membrane. So that's how they get associated with the membrane. So those are the hydrophobic sequences in there. And you can see that they are only present in 3, 4, and 6. All right, so that's it pretty much. And here, if you're just curious, I just put that additional line in the notes section. You can see that there are lots of different names of the drugs in each step. These are the experimental drugs that has been used in those. Only the monoclonal antibodies works very well. See that the chloroquine or hydroxychloroquine, if it is an endosomal pathway, it works in here blocking that. But remember that the virus can enter through the plasma membrane, so it really doesn't work in that way. On the other hand, you can see that the remdesivir is also, along with the other which are also implicated. These are the nucleoside analog. It really doesn't work very well because of the proofreading activity we are going to see in a second. All right, so this is an additional slide, which is extension of Jerry, what he told. Here you have these different membranes. So the yellow ones that you are seeing right here, the yellow ones are the double membrane vesicle, of a double membrane vesicle. So you can see the outer membrane and the inner membranes. And inside that you have these luminal filaments, and those are the replication transcription complexes and the factory to make the mRNA and the genome RNA. One of them, what you are seeing right here, is the ERGIC, Golgi Intermediate Complex as well, which is basically the area where the viruses are being synthesized. So this is different. So initially what happens that the RNA, the mRNAs and the genome RNAs are made in here, and they are exported out from here into the cytoplasm. Minus strand doesn't go out. It stays inside the double membrane vesicle. Only the plus strand mRNA and the plus strand genome RNA will go out. And this is the first time that a pore which is associated with this double membrane vesicle is associated with that. So this is the cross-section of the pore by a cryo-EM structure through which the mRNA as well as the genome RNA comes out from this double membrane vesicle into the cytoplasm. And these are the ribosomes. You can see in purple. That's going to synthesize more proteins out of those mRNAs. And these mRNAs at the late stage, what they are, these are the structural proteins of the virus. Initially, the virus comes and the 1A, 1B produce the non-structural protein to set up the replication and the transcription. And in the second set of transcription, it produces the mRNAs, which we call as a subgenomic mRNAs, to make the structural proteins. So here is a model which shows that with the help of the pore, the RNAs they're just coming out into the cytoplasm, and if they are mRNAs, they are going to get translated, and if they are genome RNA, they are going to be encapsulated by the ED protein, and then they will be destined to the ERGIC, or the endoplasmic reticulum Golgi intermediate compartments, and their viral assembly will take place. That's the last part of it. So this is basically the RTC, replication transcriptase complex, and the double membrane vesicle where the replication takes place. All right, Rachel, here? Not here? Okay. So here is the RNA-dependent RNA polymerase, some of the properties, and some of them have already been discussed. The RDRP is the core RDRP is the NSP12 and a catalytic domain, which does have this nucleotide transferase domain. NIRON is just a jargon, but this is basically where the reaction site is. And all of the polynucleotide polymerases that we have seen, DDRP, DDDP, RDRP, including RT, all of that have that nucleotide transferase domain, or the NIRON. And it is in the palm region of that right here. You can easily see that. And then what happens that the NSP7 and 8 basically forms the finger and the thumb domain, and the two NSP8 molecules produce a barrel-like structure. So here you can see that the green one is one molecule of NSP8, written as NSP8A, and this green molecule is another NSP8, which is NSP8B. Here is one NSP7, and then you have the RNA polymerase at the back right here. So what it does, that it produces a barrel-like structure. So what happens that you have the RNA template, and the RNA that is being synthesized is held in place, and it doesn't actually pull any or create a tension at the catalytic site. So that's the reason that NSP7 and 8 is called a processivity factor. Without that, RDRP can synthesize by itself, RDRP can synthesize de novo small segments of oligomers. In vitro reaction has been shown that. So small RNA, but it cannot make long RNA. If you allow them to associate it with NSP7 and 8, immediately they can synthesize very long stretches, the subgenomic mRNAs, as well as the full 30 kb of genome length RNA as well. Also, along with that, the NSP8 is basically the primase. So see that RDRP can synthesize RNA only by the primer extension, and NSP12, NSPase can synthesize RNA de novo, where the NSP8 actually acts as a primase. So that's basically what it is. So the APO enzyme is the NSP12 RDRP without process-effective factor, can synthesize short RNAs when you give them the primer. Remember that you have to give them a primer, it can only extend the primer. But if the hollow enzyme is with the 7 and 8, and 8 can synthesize its own primer, and that's your de novo synthesis, and process-effective factor that can synthesize subgenomic RNAs. And along with that, what happens that there are some other proteins like NSP14 exo-N, which excise out the misincorporated nucleotides, which is a huge big deal, and we're going to talk about that in a different slide. And this whole setup, after these proteins are synthesized in the cytoplasm, they are called the replication transcription complexes, and the RTCs, and they go to the double membrane vesicles that we are talking about. All right, so Inara is not presenting. So here, the role of the exo and the endonuclease. And the only one order, which is a Nidoviralis, and Coronaviridae is one of the families. There are 4 families under that order. And all Nidoviralis, even the other four families as well, they all have this proofreading enzyme. The reason for that the Nidoviralis, all of the members of the Nidoviralis order have the largest RNA genome. So that's the key. They all have in between a 28 kb plus genome, which is, we are going to see in a separate chapter, which should not be a proper size of an RNA genome. RNA genome after 15-20 kilobase become extremely unstable due to the huge mutation rate, because RNA-dependent RNA polymerase is a low fidelity enzyme. That means that RNA-dependent RNA polymerase makes a lot of mistake, and the mutation can make the virus dead, most of the virus particle or the virion dead due to the mutation. But that is probably the reason that they evolved to have these 2 different types of nucleases. One is the exo-N. And the exo-N works as the RNA being synthesized, mRNA or the genomic RNA. If there is a misincorporation of the nucleotide, it excises out in the 3' to 5' direction, RDRP pauses, and then continues. So here is the cause. Remdesivir is a nucleoside analog. So when it goes into the cell, it's metabolized and becomes a nucleotide, which is an analog, not exact, and it may get incorporated into the position right there. And if it incorporates, it is called a chain termination. The chain cannot get elongated. But you have the exo-N, which comes in there and excises that remdesivir out of the site. So you can have structures in vitro, structures, remdesivir binding to the binding site, and everything is good. But in physiologically, the efficacy of that goes down. So to help that issue, you have to have developed an inhibitor for the exo-N, and if you use the remdesivir along with that, that therapy may work. So that's what the people are working. So here is the structure of the exo-N, and it is NSP14, and the exo-N activity is only at its 5' side. You can see that, not 5', sorry, your N-terminal domain protein. N-terminal domain have the exo-N activity, and this MTA's activity, or the methyltransferase activity, is on its C-terminal domain. And if you're just having a question about NSP10, NSP10 is a cofactor for the methyltransferase. So that's the structure. And then the other one is NSP15. And the C terminus codes for an endo-U, or the sometimes called N-endo-U, Nendo-U, endoribonuclease, and this cuts inside a chain. Sometimes what happens that even after misincorporation, chain synthesis can go on, and suppose exo-N did not work properly, and it allowed a chain termination with a wrong nucleotide. So what will happen that in some of the areas, if there is a wrong nucleotide, suppose you have an A instead of it is base pairing with uracil or U, there is a G. So you have a bulge in the RNA duplex. So you have a bulge. So when Nendo-U is scanning that, it can scan the bulge that there is no hydrogen bonding in between the two bases. It can cut in the middle of the RNA and excite it out. And that is one of the beauty of the Endo-U. So you have two of these proofreading enzymes. So here you can see that NSP15 is the Endo-U, and the activity is at the C-terminal domain. And see that this is a typical structure of that, and you can just well imagine that this clamp-like structure just fits over the RNA, and it slides back and forth just like a clamp. So that's kind of really interesting that how it happens. And they are all potential antiviral drug targets. If you have an inhibitor for the exo-N or the endo-U, you got the key. Because if you just use the inhibitor and a nucleoside analog, it should absolutely work. All right. Hayden is not coming, she also told. So the next part is like looking at the overall machinery of the RDRP and associated non-structural protein. And just remember some of the key things. Viral genome is positive sense RNA, so it must make N- intermediate for the genome replication. And this is true for the mRNA synthesis as well. So right here, you can see that NSP12 is the RDRP. We are constantly talking, and here you have NSP7 and 8. Just only two of them are shown, but just remember that it is a multimeric protein. So now you can see that there are so many other different proteins that are associated with that. So one of them is NSP13. That's a helicase. Unwinding is being done right here. So that's one of the important part that they can do. And then you have the single-strand binding protein, NSP9, right here. And then you have the, of course, the exo-N and as well as the NSP16, which does have the 2' methyltransferase activity. You have a single-strand binding domain, NSP9. And then also you have the NSP10 is also a methyltransferase cofactor right here. So this is overall the whole of the associated proteins along with the core RDRP. And you can see 7-8 is the processivity factor, which is basically what we are calling a clamp, which is sits around the template and the strand being synthesized. All right, so after that, is Kat here? No, I got a message from her as well. Okay, so here, this is the part that I just told from her as well. Her slide is the next. So here we are looking at the genomic organization and the regulatory sequence. So what we are looking at, so far we were just only looking at the ORF1A and the B. So this is your ORF1A, and then together is ORF1B, and all your non-structural proteins were synthesized from these two, and we were only considering these two. Now let's move beyond towards the 3' end of the genome. So this is your 5' end, and this is the 3' end of the genome, and you can see that all of these are the structural proteins that we did not talk about. So all the structural proteins are at the 3' end, or towards the 3' terminal of the genome. And see that what you have, the first one is the spike. Then you have these 3A, 6, 7A, 7, these numbers. These are accessory protein with various different types of function. But the main ones is the spike protein. Then you have the envelope, E, envelope protein, M, this is a membrane protein. And then N, this is your nucleocapsid protein, which covers the RNA. So these are the 4 essential proteins. And it could be HE or the hemagglutinin esterase as well. So all of these proteins are produced from these subgenomic mRNAs, which are produced by the replication transcriptase complex inside the double membrane vesicle. So that's the overall together. But what you also see that there are some transcriptional regulatory sequence, we call that as TRS. So the first 5' end of the genome has a leader sequence, and here you have a transcriptional regulatory sequence leader. Then in between the genes, at the beginning of a gene, at the end of the gene of all structural proteins, not ORF1ab, in between the genes, you have these transcriptional regulatory sequence. We call this as a body. So all of them has these TRSBs, front and after. And we are going to see that how these are going to function basically, that basically the RDRP makes the minus strand template intermediate for the SGM RNAs at the precise 3' end of the trailer. So remember that you have a plus sense RNA. So when the RDRP first makes the minus strand RNA on the plus strand template, remember that it will start from the 3' end, and it will go, because it's going from the, so 3', here will be the complementary 5', and it will go all the way to this end. Once you have that minus strand intermediate, on top of that, it's going to get back that plus strand if it is trying to make the genome RNA. But we are going to see that there's a very unique thing happen in the subgenomic mRNA synthesis, which we call as a discontinuous synthesis. So we are going to see that how this TRS actually helps. We are going to see that from this end, the RNA will be synthesized, and it will come to a TRS, and then it can either read through the TRS, or it can jump from there all the way up to the end and finish this. So what you are going to see that all these subgenomic mRNAs will have a precise L leader sequence and a precise trailer sequence. All of them are the same, because remember that, again, I'm repeating, this is a concept which is very difficult to grasp sometimes, that it is starting the negative strand from this end. It comes in here to whatever place it just wants to, and from there it jumps all the way to the end, and it synthesizes the part. This is for the subgenomic mRNAs. So this whole part in the middle is skipped. So that is your minus template. Now when you make the plus template, you just straight go up from here, and you don't have that part, and you synthesize that part. So we're going to have multiple slides for this, just for the understanding of this. So let's go over that. So here we are looking at the Erin here. Erin? No? Okay. So here we are looking at the leader and the trailer sequence. So if you just look at the leader, you can see that there are so many stem loop structures for the RNA binding protein interaction. And here is your TRSL. And right here, you have the trailer sequence with extensive stem loop structure and some pseudoknots as well. So if you carefully look at that this is the amazing part, that right here in the trailer leader region, you have a TRS, and right here, what you are reading here, AUG. This is the start codon for every structural gene. They use that same thing. That's why I say that all the subgenomic mRNAs have the exact same leader, and the start site is that AUG. After that, the gene is specific. Then, at the end, right here, what you are seeing, this is a trailer at the end. You have an UAA. That's a stop codon. So the stop codon for every gene is the same, and after that, the trailer sequence is the same. So this is an amazing way that although these subgenomic mRNAs are small, how they can synthesize that. So if you just take out this subgenomic mRNAs, you can see that it does have this leader, and then you have a small sequence in between these sequences, not a replicase region of 1A, 1B. This sequence, then you have an S, and all the way to the end. So this subgenomic mRNA contains all of the protein in tandem, one after another, of the subgenomic RNA. The next one, it starts from here. The next one, it starts from here. And the very last one just only have this end protein. So basically what happens, the first subgenomic mRNA is the spike protein, and after that you have all other protein. But during the translation, only from each subgenomic mRNA, only the first open reading frame is translated. So you can see that it is utterly complicated, and why you have to do this, we really do not know. So basically what happens that you have all these different structures which are polyadenylated for the stability of the mRNA that is in the trailer loop, and you have lots of these bulge stem loop and the pseudoknot, and they interact with NSP8, 12 RDRP for the transcriptional regulation, because remember that RDRP will be recruited right here when it's synthesizing the minus strand, and RDRP will be recruited here when it is synthesized the plus strand from the minus template. So both of these leader and the trailers are immensely important for starting the synthesis of the genome as well as subgenomic mRNA. All right. So after that, what we are looking at, so this gives you an overview of that. So let's start from that. This is the whole thing that is coming into the cell. So you have 5' to 3', you have the whole sequence, and it translates to 1A, 1B protein. These are processed, and you get the RNA polymerase and the other thing. So now it's going to synthesize the subgenomic RNAs and the genome RNAs, and let's see what happens. So we start at the middle, not the top one. The middle is the plus all the way to the end minus. This is the genome RNA. This is the template. So when it is synthesized the genome, it will start from here. It is synthesizing all the way towards the end. This is your minus. So this top one will be the genome RNA anti-genome template. From here, it will get the whole plus back. So that's easy. Good, everyone, with that? So now the funny part happens for the subgenomic mRNA synthesis, which is the bottom part. To make the minus strand, it starts from the 3' end. We talked about that. So it goes up to this, and it hits this transcription regulatory region, which is the body, TRSB. From there, the RDRP jumps all the way towards this end and synthesizes all the way to the end. So this and this is joined together, skipping this whole middle part. And now on the top of that, when you synthesize, you just synthesize that subgenomic RNAs. And see that this is how the subgenomic mRNAs will look like. So 5' capped, 3' polyadenylated, so 2A, HES, then you have E, M, and N. So here 2A is the first protein, here H, hemagglutinin is the first protein, here S, 4, SA, E, M, and N. So what's going to happen from each of these subgenomic mRNAs during the translation, only the first protein, here it is 2A, here it is HE, here it is spike, here it is protein 4, here it is SA, here it is E, M, only the first proteins are going to be translated. And the N protein does have just only NORF, it doesn't have any other thing. So that's the way that this discontinuous synthesis basically happened. This is just a little bit of more overview of that how the open reading frame and the diversity. So I just told like what are the different types of viruses. So you can see that MHV, murine hepatitis virus, does have a hemagglutinin esterase, but none of these viruses does have that. So the main proteins are the spike, envelope, membrane, and nucleocapsid. These are the 4 structural proteins. So here is how the TRS basically is involved in the discontinuous transcription. So think about this, that the template that what you are seeing, this is your first minus template. So the RDRP starts all the way from this end. It comes to one of the TRS, and this whole structure is the RDRP complex, the purple and the green. And here, 2 things can happen at that time. So in this position, you can see that it is interacting with this leader sequence. So this is the 5' end of the genome. So this is the 3' end, this is the 5' end of the genome, and the red portion is the leader TRS, which is basically this. So this is a long distance interaction that is taking place. So here, there are 2 things can happen. Either it can stop here and interact, it can stop to the second one and interact, or it can stop to the third one and interact. So there are two things that can happen, that either it can synthesize through this TRS like this, or at the TRS, when it hits the TRS, this RNA-dependent RNA polymerase jumps to this strand from here to here and synthesize this portion. So what's going to happen, that it's going to synthesize up to this, and it joins up to this. So this whole part is skipped. And that is basically, and that can happen from here. It can come to here and jump right here. So this part will be synthesized. It can come here, and this part will be synthesized. So if it is the first one, it will synthesize from all the way up to here. So if it jumps in the first TRS, it's going to synthesize all of it. If it reads through all of them and jumps at the last TRS, then this is going to be made. So this is basically the mechanism of the discontinuous transcription, how this long distance RNA polymerase switch of template basically happens. So that's overall view of that. Then we are looking at Natalie. Okay, capping mechanism, that's interesting.
Okay, so I talked about the capping mechanism. And again, coronaviruses will use these NSP proteins to form a cap, which will allow it to be protected from the host immune system. So the first one is the NSP13, which contains that RTPase, which hydrolyzes the 5' end of the guanosine of the RNA. And then the NSP12 is associated with a GTase, which transfers a GMP molecule to the 5' end of the diphosphate RNA, which this will form that 5' triphosphate bond. And then the NSP14 contains the MTase, which methylates the N7 position of the cap, and this will form the cap 0 structure. And then NSP10 and NSP16 will form a complex and methylate the two nucleotides, which are usually G and A, after the formation of cap 0 using the 2' O MTase enzyme, and then this will be the cap 1 structure, which mimics the cellular mRNAs, which helps it to enter it.
Thank you, Natalie. Good job. So here, one of the thing that we can point out here is here you can see the methylation right here, and see that this is the second base where you have the 2' O methylation, and the third, which is in here. Remember, this is a 2' hydroxyl as well, so this can be methylated as well, the same way CH3 group can be added. And another thing, that if you just look at the slide, if you just confused with what is SAM, the SAM is the S-adenosyl methionine, which supplies the methyl group to be added. So it's S-adenosyl methionine. Methionine is gone, and that's why it's SAH comes up. So that's basically what it is. And the GTase is guanylyl transferase, and MTase is the methyl transferase. All right, great job. So after that, we are doing, Chris is not here, so we are looking at the polyadenylation. And this was the gray area for a long time. So again, the function for that is to improve the genome stability and the translational efficiency. We already know that. And the NSP8 protein in the viral polymerase has been found greatly increase the 3' polyadenylation, but it does not polyadenylate by itself. So there are some U7 tracks in there. So the earlier idea was that on the U7 tract, the RNA polymerase basically slips and adds it. This is, we have seen in VSV, we have seen it in influenza virus. They all slip on those multiple U tracks in there. But before that, the important part is that there is a hexamer, which is AGU-AAA. This is where the polyadenylation signal is. But recently, what has been discovered, that RDRP slippage by itself is not sufficient for the polyadenylation. And there is substantial evidence now that the polymerase from the cell is basically used to do this process. Now the question is that this is also, again, remember that we got this paper, it is in there. There is one huge problem with that paper. We just put it in there. I did not put it in the structure in there. Polymerase is in the nucleus. This is happening in the cytoplasm. But still remember that. There is nothing wrong, or I'm not questioning. Polymerase is synthesized inside the cytoplasm. Then it is exported to the nucleus to do the function. Somehow, the virus hijacks the polymerase from your cytoplasm to the double membrane vesicle. And that is not known. We do not know that pathway, and that is something that could be very fascinating. So that's why that we do not know much about it, but it has been shown that the host polymerase, along with the RDRP, basically polyadenylate the 3' end of the genome. So two main things. One is the U7 tract is present in there. Hexameric sequence, AGU, is a sequence for the signaling of the for the RDRP for polyadenylation, and probably the hexamer recruits a polymerase from the host as well for polyadenylation. Those are the take-home messages. So that's kind of pretty much it. So if there is any specific slide that if you just need a little bit of more detail that I can just talk about. Do you want me to go over any specific slides that you want a little bit of idea with that? Anyone can think about? So I think that this is where the most people actually struggle the jumping off it. Just remember that you have a plus strand RNA genome, you have to make a minus intermediate. And you have to make two types of minus intermediate. Either you have to make a full-length minus intermediate for the template of the full-length genome, or you made a discontinuous small minus intermediate for the subgenomic mRNAs. So when you are making that small subgenomic mRNA template, at that time, the RNA polymerase jumps. So you can see that the RNA polymerase jumps from this TRS to the next one while it is making the minus strand intermediate. When it is making the plus strand, it doesn't have to jump, because remember that this end and this end is joined together. It is just a smaller size of the anti-SG mRNA. It's just the intermediate. So it reads from here all the way. This is joined. It reads through here. So it just can go just normal way. So the jump only occurs while it makes that minus strand SG mRNA intermediate, or the template for the subgenomic mRNA. So that's basically the concept. And here also just remember that take-home message that the subgenomic mRNAs, it can have just one open reading frame, or the largest one will have all open reading frames for all the structural genes or the proteins, but only the first one from each subgenomic mRNAs, only the first one is translated. So these are some of the important ones. I guess I just underline the important ones, and then I will cover this one as well in a question-specific manner with that. So how much time do we have? So 9:06. Now is a good time to ask questions. Any questions that you guys can think of? Anything? Any related to other chapters? I will be more than happy to answer. Any things that you want me to discuss during the review or anything like that? Just you can think about. No? Everything good? Yeah, so just make sure that what I did, that each of the slides that you are going to see, you already have the slide set, you know that. You have the slide set. So I have the same slide set. The only thing that underneath that, I have the questions written in there. That's what you don't have. I have to be very careful, because it was like, if I have forgot to delete one of those slides with the questions, that could have been embarrassing. So So I'm just going to, the advantage is that when you have that material, that you can just go over one and then you can stop the video and just make sure that you got the slide and then go to the next one. And I think that even better than sitting in the class doing that, because here you are not pausing. So, and then you're in the class, then you have to rewatch the video. Here, if you just watch it 1 or 2 times, that should be great. It's going to be a difficult test. Absolutely, it will be difficult. But your lifeline is the review. I will do the review very good. So if you follow the review and understand the review, just don't memorize. Understand the review, you guys should be good. All right. Have a good break, and we will see you almost a week after, the following Tuesday.